And welcome to the Smart Science Lab about how to measure for photosynthesis and light. And what you'll notice up above here, we've got a little description of the steps of the lab. Then we've got a little guideline on what to do to measure. And then you're going to go ahead and come down here considering the central question of this lab: what is the effect of light color on photosynthesis? So whether the light is red, green, blue, or yellow, how is that going to affect the rate that the spinach photosynthesizes? You've either created or picked a hypothesis test, and then you come down and pick a color of light to examine. Let's take red. Now what you notice here is on the bottom of a dish are a bunch of discs of spinach. When I press play, the light is shining on the spinach leaf discs, and as they photosynthesize, they create some gas. That gas has the leaf disc now floating to the top of the dish. And you can measure the rate of photosynthesis by measuring how fast those little discs float to the top. So be really careful about the way you measure this. This is a little different than some of the other labs. You have to make sure and notice whether or not the disc has floated. So they start all at the bottom. We press next. And as we press next, time is advancing. Now, once we see the first disc lifting up to the top, there it is, click on it. That will then get the whole graph started, because if there was zero data, they weren't measuring those points until you finally get that first point clicked on. So you'll notice that each next that I'm pressing is 1 minute or 60 seconds of time. And each time a disc floats up to the top, I'm just going to go ahead and click on it and mark it. And so you're going to see that you can advance time, you can mark discs, and eventually all the discs are going to have floated up to the top, and you're going to measure all of them. Once that's happened, then you'll get a chance to save your data with the save button on the right. Let's do that right now. Once you've saved it, you can see, bam, there's our red color in red. And what you want to do is do all four of these different colors, compare the graphs, and see which one had the fastest rate of photosynthesis, or in other words, the fastest that these discs floated up to the top of the dish that they're in. The discs float due to gas being created during the process of photosynthesis. Thank you so much, and do well with your photosynthesis and light lab here with this Smart Science video.